During development, vertebrates undergo a process called neurulation, which forms the neural tube, this structure, and it involves two important proteins called BMP and SHH. BMP stands for bone morphogenetic protein, and SHH stands for sonic hedgehog. During this process, the ectoderm here contributes to the formation of the roof plate by secreting BMP. At the same time, the notochord, this structure, contributes to the floor plate by secreting SHH. After the ectoderm and the roof plate are formed, they both release BMP downward in this direction. At the same time, and opposite to the BMP secretion, the notochord and the floor plate secrete sonic hedgehog in the upwards direction, which contributes to a particular gradient formed here. Our proposal is based on previous research that has shown that sonic hedgehog coming from the notochord is sufficient. In other words, we don't need any SHH from the floor plate in order to get the gradient. What we want to see is the same thing, but for BMP. BMP is released from both the ectoderm and the roof plate. We want to see which source is sufficient towards establishing the gradient.
Mutations in the SHH gene are the most common cause of sporadic and inherited holoprosencephaly, a developmental disorder that is characterized by defective prosencephalon development. The SHH transcription pathway has also been implicated in the formation of specific types of cancerous tumors, including embryonic cerebellar tumors, medulloblastoma, as well as the progression of prostate cancer tumors. Similarly, mutations in BMP have been linked to fibrodysplasia, a disease involving skeletal abnormalities. This investigation aims to gain insight into the role of morphogens in the development of the neural tube and perhaps aid further studies in shedding light on the pathologies of these disorders.
Specifically, our research question is stated as follows. Is ecto-BMP or RP-BMP sufficient for the dorsal patterning of the neural tube? We will test this through three hypotheses. 1. Ecto-BMP is sufficient for regular dorsal vertebral patterning. 2. Roof plate BMP is sufficient for regular dorsal vertebral patterning. And 3. Both ecto-BMP and RP-BMP together are required for dorsal vertebral patterning.
So our first hypothesis is ecto-BMP sufficient for the regular dorsal vertebral patterning? The materials we used for this experiment are homozygous floxed conditional RP-BMP mouse embryonic fibroblast model, tamoxifen, and an RNA probe. Using a similar method as outlined by Choi et al. 2012, we will generate a homozygous LOX-P flox tamoxifen conditional RP-BMP mouse model. We will then restrict the expression of BMP to the ectoderm through temporal administration of tamoxifen, thus silencing RP-BMP. We will then build a fluorescent probe using a sequence complementary to the floxed region of BMP to visualize the localization of BMP mRNA. We will then stain and fix the sample when complete. If hypothesis 1 is true and ecto-BMP is sufficient, histological analysis will show that the embryo has proper dorsal ventral axis formation and normal vertebral column patterning.
Our second hypothesis is that RP-BMP is sufficient for regular dorsal ventral vertebral patterning. Our materials will be using our CRISPR-Cas9 gRNA or guide RNA and a fluorescent probe. So using a inducible TET CRISPR-Cas9 system, we will design a gRNA to target and silence ecto-BMP. Following administration of TET, Cas9 will be activated and cleave ecto-BMP, thus knocking it out. We will then build a fluorescent probe complementary to the BMP RNA, then observe the localization of its fluorescence. If hypothesis 2 is true and RP-BMP is sufficient, histological analysis will show that the embryo has proper dorsal ventral axis formation and a normal vertebral column patterning. Our last hypothesis states that both ecto-BMP and RP-BMP together are required for regular patterning. The materials we'll be using are the materials used in previous experiments used to knock out ecto-BMP and RP-BMP. Furthermore, we'll also be using chemically synthesized siRNA. We'll be using this sRNA to target and silence both ecto-BMP and RP-BMP. At this stage of the experiment, this is equivalent to a negative control, meaning that no BMP mRNA should be visualized. Utilizing methods from experiments 1 and 2, we will knock out ecto-BMP and RP-BMP individually and visualize BMP mRNA with a fluorescent probe. We will also include a positive control in which ecto-BMP and RP-BMP are expressed regularly. If hypothesis 3 is true, then we'll observe normal vertebral column patterning and proper dorsal ventral axis formation only when both ecto-BMP and RP-BMP are present together.